The ability to create a clone used to be science fiction. Since then, the debate on applying the technique to clone human beings has been ongoing. Based on all the theories and discussion, it may sound like human or otherwise is an easy process. Find out what actually involved in the cloning process by viewing this presentation. In this video presentation, we are going to review the key concept of what is cloning, the history of cloning, the types of cloning, what is organ cloning, advantages and disadvantages of human cloning, religious insight about organ cloning, and the future of cloning. We are going to begin this presentation by identifying what is cloning. The word cloning simply means copying. When people think about cloning, they usually think about complete human beings or animal life. But cloning is also used for purposes other than making exact copies of animal or people. It is a process of generating a genetically identical copy of a cell or an organism. Also, it is the act of reprogramming a cell by replacing its nucleus with that another cell, so it becomes the genetic equivalent to the original. The history of cloning began in 1952 when Robert Briggs and Thomas J. King was successfully cloned tadpoles by a nuclear transfer. It is the world's first ever successful animal cloning experiment in the history. In 1996, the world's first ever cloned mammal named Dolly the sheep, the first animal clone from an adult somatic cell. In 2004, South Korean scientists create several cloned human embryos, which they only allowed to develop for a short while, just enough to extract embryonic stem cells which could be used in the treatment of any diseases. There are three types of cloning: the DNA or the gene cloning, the therapeutic cloning, and the reproductive cloning. When we say about DNA or gene cloning, it refers to the method of replicating one molecule to produce population of cells with identical DNA molecules. While the therapeutic cloning contains the genetic material from any cell of the body and it's transferred by injecting into an unfertilized egg from the nucleus, which has also been removed. Lastly, the reproductive cloning is a process of creating an animal that is genetically identical to a donor animal through a somatic cell nuclear transfer. At this point, we are going to identify what is organ cloning. Organ cloning is the creation of a genetic identically copy of human organ. It is a process used to create an exact genetic replica of another cell, tissue, or organism. On legal basis, Section 5 of the Human Cloning and Other Prohibited Practices Act of 2004 prohibits placing human embryo clone in the body of human and animals. In terms of Section 39A of the Human Tissue Act 65 of 1983, genetic manipulation of gametes or zygotes outside the human body is absolutely prohibited. What are the advantages of human cloning? First, a defective gene could be eliminated by healthy organ cloning cells. Also, faster recovery by a traumatic injury by cloning cell to help the recovery process. And lastly, infertility could be eliminated. In contrary, formation of new diseases may occur. Also, Organ rejection varies because of cell mutation. And lastly, the uncertainty. Many of the effects of cloning are unknown. It is a new world of science that is still continually being discovered. The religious objection to cloning is still in process. Many religions oppose human cloning and the cloning of human embryo, since they believe that life begins at the moment of conception, and the soul gets into the body only this way. When we talk about future of cloning is a popular topic of discussion. The future of cloning looks very bright as the scientists are constantly developing new methods. However, it is not quite sure whether the future of cloning will help humans to be the cause for its destruction. In conclusion, organ cloning is unsafe at this time because the complete list of defects can be accounted for. The sacrifices of countless children to death and physical abnormalities, as well as mental illnesses, it can be blamed for the sake of science. However, the future of cloning looking forward for the new set of ideas, future studies, highly invented machines, and the most important is the dedication of the future learners for the success of their experiment. That's all about cloning and organ cloning. Thank you very much and have a great day.